Hello, in this video we will discuss about the human endocannabinoid system. So ECS, we will discuss what is that, and the basically is the general overview. If we will discuss about the endocannabinoid system consists of cannabinoid receptor, endocannabinoids, chemical, and their metabolic enzyme. So there are three types which that is used for the endocannabinoid system in the human. It is regulate and homeostasis, and it is very important for the human normal activity. So the endocannabinoid receptor is basically, first of all we will discuss is the two types, the CB1 receptor and CB2 receptor. While the endocannabinoids chemical or endogenous chemical which that is present in the human body is known as the anandamide AEA and arachidonyl glycerol 2AG, so as well as enzyme for degradation. So here is the nervous system we will discuss also, but here is the exogenous cannabinoids can be phytocannabinoids from the marijuana to tetrahydrocannabinoids THC and the cannabidiol which that is the CBD. This is the two type, and there are 1,000 type of the cannabinoids are present in the different plant. While these plants contain two types, a major. Well, let's begin to understand with the other things. Here is the endocannabinoid system is the discovered in the 1990, and exogenous cannabinoids bind with CB1, CB2 receptor with great specificity, much like lock and key. So the specific cannabinoids will bind with a specific time and the specific site for the inhibition of the emotion. So here is the different type, and there is the location of the receptor can be throughout the body. But important thing is that the receptor CB1 receptor basically is cannabinoids receptor are widely distributed throughout the human body, but the receptor CB1 receptor are mainly located in the brain and central nervous system, the brain and spinal cord, but are also found in other tissue. There are a lot of tissue, for example pancreas and other liver, etc., for regulation and homeostasis. But the changes of the environment if we will provide the exogenous cannabinoids. So here is the spinal cord and brain. So let's begin to understand about the activation of cannabinoid receptor to inhibit the release of neurotransmitter. So the neurotransmitter which that is released from the presynaptic neuron to the postsynaptic neuron for the conduction of muscle as well as the hormone production or the digestion. So there are the different types. Here is the CB1 we will discuss. If so, the AEA will be bind the naturally inside the endogenous, and the 2AG. Basically, this is the ligand, and this will bind with the CB1. That CB1 are present in the nervous system, nerves, neuron. So for example, here is the CBD, the cannabidiol, which that is the exogenous, can be also bind from this region. But here is the CB2. This will bind with the this is the receptor will bind the THC, that is the trihydrocannabinoids, and it will be bind within it, and it can be also AEA and 2AG. So the CB2 receptor are most densely found in immunological tissue and modulate cell fate. So the proliferation, etc., there are a lot of things which that is happening. For example, the inhibition of the inflammation also. So the ECS play key role in the homeostasis and regulates many physiological processes such as inflammation and pain regulation, perception, immunity, and neuropathy and metabolism. So let's begin to understand the synapse. So here I am drawing the nervous system, which that here is the afferent and efferent neurons, which that is the presynaptic neuron is we will discuss when we will smoke. So the cannabinoid will move into the blood circulatory system. After the moving into the blood circulatory system, will bind with the cannabinoid receptor, where the present here is the neuron, which that is the presynaptic neuron, and this is the neurotransmitter will inhibit by the THC binding exogenous cannabinoids, while in the case of other endogenous can bind to trigger more regulation and more stimulation to exocytosis. Fire here is the neurotransmitter receptor, and this is used for the reception, and this is the post neuron, which that give the signal to the body, for example brain to the muscle and muscle to the brain. So the AEA bind to the CB1 receptor with great affinity than CB2, whereas 2AG binds both receptors with equal affinity. So in this way, the CB1 receptor will bind great, AEA bind with great affinity with the CB2. The THC binds the CB1 receptor with great affinity than the CB2 receptor, and it has been suggested the binding effect of the THC mimic AEA. And CBD has low affinity for both receptors, at low concentration. It has proposed that binding effect of CBD are mimic to 2AG. So in this way, the affinity for both receptor is equal CBD for, due to the increasing of the CBD can increase the increase the affinity of the nervous system for binding to inhibit the stimulation of nerve impulse, and therefore this is the exogenous. So that is why here is I am drawing the bigger picture for understanding the synapse. So the presynaptic neuron and this is the postsynaptic neuron contain receptor, while the vesicle are present in the presynaptic neuron, and there are here you can see CB2 receptor and CB1 receptor can bind with the THC and AEA and THC and 2AG. This is the exogenous and endogenous cannabinoids, and here the neurotransmitter receptor are present, which that is receive and give the signal, and after this here is the lipid precursor will become a 2AG and AEA. So this is the basically the 2A arachidonyl glycerol, and as well as the amide and amide, so will bind, so to give the triggering or inhibition on the basis of that is why we can lose memory during the one consumption of the exogenously. So here is the exogenous can be THC, tetrahydrocannabinol, and the endogenous is the N-amide and 2-arachidonyl which that is a chemical formula you can understand as well as. So in this way, the exogenously THC can be change their shape after the smoking. So during the burning time, it can be change their structure due to the heat. After the heat, it can be change their structure to give the different function with the different things you should know about that. And here is the phosphatidyl ethanol amine, which that is convert the biosynthesis we will discuss here. So this is the N-acetyl transferase enzyme used to N-arachidonyl phosphatidyl ethanol amine formation will be occur, and in this way the phospholipase D, which that is used for the N and amine formation, and this is the basically the cannabinoid, while the fatty acid amide hydrolase enzyme used to ethanol amine formation and arachidonic acid formation. This is the degradation. So after this degradation can be again reused. Ethanol amine is used for another mechanism, and arachidonic acid which that is used for another mechanism. So on the other hand, here is that was the N and amide AEA was, but here is the phosphatidyl inositol convert into the phospholipase enzyme presence phospholipase C and diacylglycerol, DAG, which that is converted into the DAG lipase enzyme used to it to produce the 2-arachidonyl glycerol 2AG. This is the cannabinoid. So remember about that. This is the endogenous formation of the biosynthesis, while the plant will synthesis outside in the plant basically through a gene formation. So the expression of the gene more in the marijuana. So the monoacylglycerol lipase is used for the conversion into the glycerol and arachidonic acid. So here is the binding with nervous system with the two endogenous cannabinoids with gland, immune cells, organs, brain, connective tissue working to handle the many functions necessary for survival. So the homeostasis can be the sleep, appetite, pain, inflammation, memory, and mood, as well as reproduction, which that is controlled by this system. So the ECS is very important. And here is the 2AG and AEA, which that is the different endogenous cannabinoids, which that in this way the appetite, pain response, and immune system function and through 2AE, while the AEA is used for the bliss molecules and the runners and high bliss full state. During the happy time it will be released and bind to increase the happiness. So thanks for watching. Please make sure to subscribe, like, and share. Ask the question for answer and comment about the artwork.